Um, yeah, okay. Yeah, 12:20, I think we can start, right?
Yep, we're ready.
You ready? Okay. So hi, everyone. It's my pleasure to introduce Drew Harner. Drew is a PhD candidate in the department in the horticulture program. So Drew has been around for a while. He has first he started his master in 2016 in our department in the viticulture lab. And after finishing his master in 2018, he started his PhD still in the same program and here at Penn State. And we are happy he decided to keep working with grapes and here in Penn State. So Drew, for his master, his master work was about evaluating the effect of weather and also cultural factors on wine grape aroma, so aroma that are important for wine quality. And for his PhD work, he kind of broadened his research interest. So he's still working on models, so how to predict wine important flavor for wine quality through models that incorporate weather and cultural parameter, but also has been working on a novel pest or insect pest, which is the spotted lanternfly. I guess doesn't need an introduction, but I mean, it's been a challenging or big problem for grape grower, especially in Southeast PA. So I'm glad Drew decided to to work also on better understanding the grapevine response to spotted lanternfly. So today he will give us an update on his research project, which is a big collaboration of many also people. And this is not his exit seminar, just an update that he offered to give to the department about this project. And with this, I will turn it over to Drew. Thank you, Drew. I will mute myself.
Thanks, Michaela. So hi, everybody. Thanks for tuning in today on this Friday. As Michaela said, this is going to be kind of an update on one part of my PhD program, specifically on a project that we began in 2019 that's still ongoing. And as Michaela mentioned, this was a big collaborative project, or it still is, involving many people across the college, as well as some colleagues in the entomology department. So just big shout out to all those people for help making this happen already and giving me the opportunity to work on this. So to get started, for those of you who have yet to see a lanternfly or have escaped all the notices and newsletters about them and haven't seen it, this is what they look like in the upper corner. This is an adult female spotted lanternfly. And just to give you kind of a background on them, they're indigenous to southern China. They're in the Hemipteran order, so they're related to other kind of familiar insects like aphids and cicadas and other insects like that. They're phloem feeding, so they feed directly on the plant phloem. They have this little stylet that just pierces the phloem, and then they just suck the sap right out of that. And they were introduced to Pennsylvania in 2014, down in southeastern Pennsylvania, outside Philadelphia. And some entomologists think that they've actually been in the state since 2012, based on kind of older egg masses that have been found. But they really began in the southeast PA. And this is actually the recently updated map of their kind of current distribution across the Northeast. And you see that since their introduction, they've expanded throughout southeast PA into central PA. And then there's isolated populations in western PA, as well as eastern Ohio, and then, surrounding northeastern states. And there's been individuals popping up all over the place, even up in the Finger Lakes, but to date, these are really where the established populations are. And in terms of the threat they pose to plants, they can feed on over 70 documented species of plants, which is quite a bit, but their preferred host seems to be tree of heaven, which is another introduced perennial tree species. And they also seem to really like grapes, as we've come to know. And in terms of economic damage that's been reported due to lanternfly infestations, it seems to be very strongly represented in vineyard systems. with damage being reported by growers as recently as 2017 to 2018 was really when it first started picking up. And growers were reporting a variety of things, but mostly like a general vine decline, decreased yields, decreased fruit quality. And this is both alarming from obviously the industry perspective as well as to us, and poses all sorts of questions because this is a novel insect pest to the Northeast. And there's very little research and information available to us on how plants respond to spotted lanternfly feeding and what they can really do to plants in high numbers and high infestation numbers like we're seeing in some vineyards. So we're really in the dark. So when we're trying to figure out how to even manage this, we're just trying to even understand what they even do to grapes to begin with. And when I mentioned high, and you'll see actually in this photo on the bottom left is them perching on some grape stems. And when I mentioned high infestations, these are just some photos from the field from Heather Leach and Michaela. And when I say high infestation, I mean like they're congregating on vines in very high numbers and feeding on a single vine. And in vineyards with really bad cases, it's possible to see this many, and they're distributed on the trunks, but also up into the canopy on the shoots as well. So we're really curious about, when you have this many lanternflies on a single vine, like what are they doing as they feeding to it? Because that's quite a lot of insects and quite a bit of an infestation. And I think the best way to approach that and try to conceptualize what they can do is really to think about their life cycle in relation to the grapevine kind of seasonal phenology. So lanternflies only have one life cycle per year. They hatch back in around early June is when they start, late May, early June is when they start emerging from their egg masses, and they start as these tiny little instars. And while they're hatching, you'll see down here is kind of really perhaps simplistic, but still a good representative example of how grapes grow through the season. And when lanternflies are hatching, it's when grapes are really in their most intensive period of shoot growth. So it's right around bloom in early June, and this is when most of the vegetative biomass is really accumulating and growing. So at this point in time, growers have a comparatively easier time managing populations compared to later when they're adults, because when they hatch, they don't have wings. So their mobility is restrained really within the areas in which they hatch and where their egg masses are. And in terms of really chemical control is what I think would be the quickest option to removing their populations. So they're not as much of a threat at that point in the time of the year, but as the season progresses and as the grapevines continue to grow and then grapes begin to ripen, they end up beginning their ripening about the same time as the lanternflies are emerging into adults. And this is really when we start to see, I guess, a phase of the season in which they potentially could have more direct effects on the vine themselves, because they're bigger and they're just feeding in larger numbers, but also they have wings and they can now fly and relocate between vineyards. So you tend to see populations moving a bit and moving into vineyards at this time concurrently while grapes are starting to ripen. And this poses a series of problems, mostly because as you go through the ripening period and as you approach harvest, growers have increased restriction on what they can spray in terms of insecticides due to pre-harvest intervals, as well as there's potential complications between lanternfly feeding and other physiological processes happening within the vines themselves. Obviously, they're ripening. Fruit quality is important, and clusters and grapes at this point are really strong sinks. So there's potential interplay there that I really think is the best conceptual way to understand how we're approaching our research, and or at least for what I'm presenting today, as well as really the potential threat they can have to vineyards. Because as I mentioned, once you get into the ripening period, grapes become a big sink. And in terms of source-sink relationships, very important for fruit crops as a whole. But during this period, from about early to mid-August up until ripening and harvest, the grapes really become strong sinks for a lot of things, including carbon and nitrogen. So on the left, this is just a brief diagram of kind of what's happening at this ripening period. On the left is kind of my carbon diagram, and on the right is the nitrogen. And essentially the gist is that as grapes are ripening, any carbon that's being fixed into the canopy is most likely being directed to ripening fruit. But also at this time of the year, grapevines are simultaneously beginning their physiological processes related to cold acclimation and kind of nutrient storage for next season's growth. So you get this overlap of both fruit ripening and then winter hardiness beginning, preparing, as well as winter reserves. And then same thing with nitrogen. The sink or fruit is a strong sink for nitrogen, both nitrogen being taken up by the roots as well as nitrogen being reallocated within the canopy and the plant itself. So you get this strong sink strength between fruit as well as reserves, where carbon and nitrogen is accumulating around this time of the season. And obviously there's implications there for fruit quality. We need sugar in grapes to ferment for wine. So sugars and other carbon-based molecules are very important for various wine aromatic chemistry, as well as nitrogenous compounds. And then also, winter reserves and other compounds related to winter hardiness, as well as early season growth is important for the next year. So then the question then becomes, well, what happens when you put a lanternfly on a vine? And particularly something that requires a high amount of both carbon and nitrogen for its dietary needs and sap. As I mentioned, lanternflies are sap feeding, so they require quite a large volume of sap in order to meet these dietary requirements. So there's the potential that they're exerting a sink strength. And with this comes the question, well, even in terms of like fruit load, how does different fruit load and cropping load affect sourcing relationships? Again, and for a given vine, if you're all of a sudden introducing a whole host of lanternflies that are feeding and removing carbon or exerting sink strength, how does that impact other aspects of the vine, like fruit quality and ripening, as well as those reserves? So then related to this, then arises the question of, is there a threshold at which grapevines can grapevines withstand infestation levels up to a certain point at which they either begin to lose nutrients to lanternflies, or nutrients stop going towards the fruit or reserves and then are just almost exclusively allocated to the lanternflies due to the sink strength of the infestations? And then that's really the question that we have is, how do different infestation densities, affect these source sink relationships? Because in terms of a grower's perspective, if they have a vineyard and their grapevines can withstand up to 100 lanternflies per vine, for example, that's just as an example. And the grapes can still ripen, they can still get the desired fruit quality, and grapevine health is not impacted, then there's no necessarily a need to spray until you hit that point. So there's an economic perspective here that's very important for growers that we're also trying to get at. So with this kind of thematic conception in mind, I wanted to introduce my research questions. So again, we were really curious about this population density effect and this vine decline that growers are reporting and these other effects. We don't to date have a mechanism for really what's going on. And because this is such a novel pest, it's given us the opportunity to really probe different aspects of grapevine physiology and functioning to really see what's exactly happening in response to lanternfly feeding. So my first question deals with both physiology at the leaf and the whole vine perspective. So essentially gas exchange versus like transpiration and whole vine sap flow. And then our second kind of targeted question looks at primary metabolites, namely carbon-based and nitrogen metabolites, because again, both of those classes are obviously important for plant health, but also long-term functioning, as well as grape quality and wine quality. And then when you're considering these two questions, there's a third question that pops up too, that grapevines are perennial plants, as well as many other fruit crops. So what you do in one season could potentially have an effect in the next season on vine health or vine quality. And that's the question a lot of growers have, and we also have too, is, when you're seeing these repeated infestations year after year, what's the cumulative effect on a vineyard or a vine's health? So that's the third question we have is, what are the effects on the carryover kind of qualities of the grapevines. And you'll notice that I don't mention anything about the fruit in here per se yet. And we did harvest fruit, and we do have some measurements we're doing on the fruit that we harvested, but the vines that we had to select for our experiment didn't have representative kind of standard crop loads. So the results that we could yield from that, we're waiting to get more years of data that are more representative of typical years, from which we could have better idea of what the effects are in fruit. But so with these questions in mind, our experimental design, I just wanted to go over real quick. Our experiment took place in southeastern PA within the quarantine zone, kind of near Allentown area. And we found a grower who was willing to let us infest his vines with lanternflies, which we're very, grateful for. And we took 16 Riesling vines and built individual cages around each that we were essentially going to introduce known populations of lanternflies into. And for this experiment, again, we only used adult lanternflies because during the ripening phase, those are the ones that are present. Those are the ones that are present in the highest amounts and are feeding the most. So for us, it made the most sense. And then also in terms of our infestation cycles or infestation processes, what we did is we set up what we called infestation cycles beginning in late August when ripening is in full force, all the way up into mid-October, which is actually in the post-harvest kind of period. And what we did was we went out, we collected lanternflies, and then came back and introduced known amounts of lanternflies into each cage. And we exposed these vines for 3 to 4 days. And then we pulled them, went in, counted all the lanternflies, removed them, and then gave the vines a 3 to 4 day no infestation period. And we did this because we wanted to replicate what would be happening population-wise in a normal vineyard setting, in which growers would be going in and spraying insecticides and essentially knocking down populations. And then a couple of days would pass before vineyard populations would begin to pick up again. So we were trying to replicate a commercial setting while still having a controlled design experiment. So then in terms of our treatments, you can see the cycle dates on the bottom, but we had 4 treatments set up in a randomized block design in which we had control vines with no lanternflies, and then a scaling treatment, set up in which we had 50 lanternflies per vine were our low infestation densities, and then our medium and our highs were 100 lanternflies per vine, and then 200. So we really tried to have a big range so we could really see, what really happens to the grapevines when there's high infestation, high repeated infestation densities. So to get more into some of our methods and then ultimately our results, my first question really delved into grapevine physiology. And for leaf physiology, we focused mainly on gas exchange, leaf gas exchange, specifically carbon assimilation, leaf transpiration, and stomatal conductance, just using a portable gas exchange analyzer that you see me carrying on the right. And for this, we targeted the upper canopy and the leaves that were young leaves, but essentially the most photosynthetically active. And we did this because we wanted to have a representative sample of the canopy of where photosynthesis is really occurring at the highest at this point in time. And also just to get a snapshot of, what lanternfly infestation density can do to gas exchange parameters. So then if you transition from the leaves, we also used a variety of whole vine measurement approaches, namely using 2 different technologies. The first were sap flow sensors. So these are a bit more intuitive, but essentially they're sensors that you install on the vines, on the trunks, that measure sap flow or transpiration. And we have trunks or vines that have 2 trunks. So we have 2 sap flow sensors per trunk, which is what you see in this photo beneath all this insulation. And this is what the sensors look like. They're just a heating pad with thermocouples that you essentially just wrap around the trunk after you've prepped the trunk. And then the way they work is that they heat up on one side of the sensor, they heat sap, and then as sap moves past that sensor and the thermocouple, it's heated, and then the thermocouples at the top of the sensor measure the sap again. And then that difference in temperature can then be used to calculate essentially the sap flow velocity. So this gives us a continuous measurement of whole vine transpiration. We installed these late in July, so we have data from that all the way up until late October. So it gives us a nice continuous look at transpiration patterns. And then we coupled that with point dendrometers, which we installed on the base of the vines. And I'm going to be talking more about these in the next couple slides, but essentially these, when coupled with the sap flow sensors, these allow us to look at diurnal and long-term trends in kind of the trunk variation. They measure the trunk radial growth as the trunk expands and contracts. And we also from these can get xylem and phloem specific data. So these coupled with the sap flow sensors really give us a nuanced, continuous, experimental wide look at really how the vine is responding to the lanternflies as we're infesting them. So then just to speak more about the point dendrometers for a second, I was more unfamiliar with these as a tool for research, but they tend to be used more in ecological and like forestry based systems. But they're really cool because with them, it's a super sensitive sensor. You just kind of rest right on the bark if that's where you want it. And then it essentially just picks up the displacement as the trunk expands and contracts within a given day due to, transpiration and other photosynthetic kind of source sink relationship factors, as well as in long term. In a given season, as the trunk grows, it can pick up trunk growth. So you really get this dual look at long-term patterns as well as an ability to look at short-term patterns. And the ones that we have actually are two sensors in one. So this diagram on the right kind of gives you a brief rundown. And this is xylem tissue, and this is the ring of phloem, and the cambium with the bark on the outside. And one of our sensors was positioned on the outside of the bark, which would give us the whole variation from the trunk itself. And then we also went through and removed a small square of phloem and put the second point dendrometer right on the xylem itself. So we also got data from xylem expansion and contraction too. And then when you subtract the xylem one from the bark one, you can get the phloem data. So I was really excited about these sensors because they really give us the ability to look at xylem activity and phloem activity independently, as well as in tandem with the whole stem and see what's contributing to stem variation. And then once you add the sap flow data onto that, we get a pretty complete photo or complete understanding of kind of the water dynamics and the phloem dynamics going on in the vine at that time. So then to get into kind of our hypotheses and what we were thinking, what we were going to see, with this scaling infestation density treatments, I really thought that we'd see a density dependent responses such that as you added more lanternflies, you would see increasing responses for various variables. So for example, for leaf gas exchange, based on the literature I've seen, there doesn't seem to be a compensatory response in response to sap feeding insects as compared to insects that directly remove leaf biomass, like a caterpillar or something. So I really expected leaf gas exchange to go down as we added more lanternflies with an increasing effect with that scaled with the density. And then coupled with that, if we were getting this depression of leaf gas exchange, you would think we'd also have a concurrent reduction in whole vine sap flow or transpiration because the leaves are simply not transpiring as much and there's less assimilation going on, and the stomates aren't open then. So then coupled with that, in terms of the dendrometers, I also thought we would see a gradual and an increased contraction of the trunk. And to give a quick explainer on that, when measuring these long-term and short-term trunk trends for radial growth, in other fruit systems that dendrometers have been used, long-term drought has shown to kind of decrease the trunk radial expansion and actually make it contract. And also a couple girdling experiments that I was reading about also tend to have the effect of halting growth and potentially in the long term leading to a contraction. So then when we're in this system in which we have lanternflies that are both physically removing a lot of water from the vines as well via sap, as well as photoassimilates that contribute to osmoregularity between the tissues, I thought we would really see this long-term gradual shrinkage of the trunk. That's a bit more complicated, but that's what I was thinking. And then when you look at the data, so this first graph is our carbon assimilation data, so the gas exchange, or one component of the gas exchange data. And you see on the x-axis, I only have 4 cycles represented. We had 6 infestation cycles. But by the time we got past cycle 4, it was early October and it was too rainy and cloudy to be able to take accurate data. But even within these 4 cycles, if you look on the y-axis, this is carbon assimilation expressed as percent difference from control. So that's why there's only 3 bars, each corresponding to a treatment. And what you see is that as time progressed and as we added repeated infestations, we saw an increasingly strong reduction in carbon assimilation based by treatment. So by cycle 2, we saw a significant reduction already with the high treatments. And then cycle 3, we started seeing significant results with the medium. And then by cycle 4, all treatments were significantly reduced in terms of carbon assimilation. And the leaf transpiration data, as well as the stomatal conductance data, are exact mirrors of this. So there was this overall trend of really strong, quick suppression occurring with higher density. But then by the end of the experiment, all the treatments were responding with some kind of suppressed carbon assimilation. So then when you look at the sap flow data, and this is kind of a confusing graph or complex, so I'm going to work through it with you. But again, on the x-axis is our cycles. So all the infestation cycles, including the baseline kind of pre-infestation period. And then on the y-axis is our sap flow. And I have the sap flow expressed as an average daily flow rate per cycle. So each cycle, again, was 3 to 4 days. Each day I would calculate an average flow rate for that day, and then I average all those for the cycle. So that's why there's only one value being expressed per treatment per cycle. And then all these values are standardized to the leaf area of the vine in order to account for different vine sizes. So then to jump into it, one challenge we had right off the bat with this data set was that if you look at the baseline pre-infestation data, one of our treatments, the high treatment, is already expressing a significantly high sap flow compared to the other treatments before we even did anything. So this was a bit of a challenge because it caused us we kind of had to address it with various statistical methods. So after talking with Rich Marini a lot, we decided to include all the baseline data in our analysis as a covariate in order to account for all the variation we were seeing in the early part of the data set. But that being said, this graph is simply just the means per cycle. So this isn't like a means adjusted graph, just for clarity's sake. But some trends still emerged, namely that as you go through the experimental phase, you see that for cycles 1 through 3, most of the treatments were tracking along together pretty nicely. We were starting to see a reduction in the high vines already. But then really it's by the end of the experiment that you see this grouping emerge, such that the control and the low vines stayed together. And then the medium and the high were pretty similar in their sap flow rates too. And then once we went through and did all the statistical tests, by the end, we were seeing that the control and the lows the rates of the control and the low vines were significantly different from the high vines. So again, we saw this long-term kind of reduction in physiological activity with the highest infestation density, which was something that we thought was interesting, and also that the lower vines were pretty much the same as the control. So that's also worth noting too. But again, lots of lanternflies were still leading to some level of reduction in sap flow, which matched the gas exchange data. So then when we pivot and we think about the dendrometer data, it kind of requires a little bit of a change of mindset, because we're talking more about radial growth and radial expansion of the trunk. And it's important to note that grapevines have a slightly strange seasonal pattern of growth to begin with, because generally, intuitively, when we think of growth, we think of it as this, presumably linear process across the growing season, and then growth stops. Well, with grapes, what happens is that lateral secondary growth on trunks doesn't actually start until after bloom. And so then this is a graph of just one of our vines, and this is on the x-axis is the date beginning in July and ending past November. And then on the y-axis is the trunk diameter, standardized to the beginning of the growth period. So everything is in relation to, how wide the trunk was when I first started this. And what you see is that, trunks, the grapevine trunk grows as the season goes on, as is expected. And this is a period of missing data, just FYI. But then all of our vines, we saw that growth increased up into about here. And this is essentially where ripening begins. And with grapevines, as I mentioned, once ripening begins, a host of other physiological processes start for long-term cold acclimation as well as next season storage. So what we see, this reduction is actually, based on literature that I was reading, is due to both the reallocation of photoassimilates away from growth towards ripening, as well as trunk contraction that's related to kind of an anatomical preparation for the winter and for essentially like cold hardiness related anatomical changes, which is cool, but it makes the data kind of strange to look at because that arrow is where our experiment began, and already we have this like negative trend we have to account for. So it's a little bit of a different data set to look at. But if we were to look at our experimental period, this is the whole thing graphed out. Again, dates on the x-axis beginning with grapevine ripening and ending a little after November. And then on the y is the trunk diameter, standardized to the beginning of the ripening period. And then in terms of the lines, the blue is the control, orange is low, gray is medium, and the yellow is high. And the arrow is where the experiment begins. And the important thing to note here is that, we can quantify all sorts of metrics for diurnal characteristics using these dendrometers. But for the sake of this experiment, I just was looking, not this experiment, this presentation, I was looking at long-term trends. And what you see interestingly is that as the experiment continues, we see a net contraction with the medium, this gray line, and the high treatments, these yellow lines, such that by the time the experiment ends and we begin to get into really late season senescence kind of phenology, these vines have actually shrunk, their trunks have shrunk. And this was interesting because I didn't think this was gonna happen I thought it was gonna happen for all the treatments, but again, like the safflow, the low vines ended up actually sticking with the control vines for the duration of the experiment, whereas the medium and the high experienced more of a decrease. And this is something we're gonna have to parse into more. But like I mentioned before, these contractions are typically seen under drought scenarios, which is interesting to me because this would then suggest that maybe the removal of water by the lanternflies is potentially approximating something similar to kind of a drought condition, but we never saw any visible drought symptoms on the vines themselves. And also, again, this is the whole trunk. So I do have the xylem and phloem specific data. I don't have it in this presentation, but that's really the next step, because then by looking at that, we can see, well, when compared to the control and the low vines, these medium and these high vines, is it the xylem that's shrinking and contributing to this shrinkage, which would be uncommon because in most cases it's the phloem that shrinks the most in drought conditions. And again, lanternfly being a phloem feeding insect would lead me to think that it would be the phloem, but again, this is something I would need to look at and verify myself. But yeah, this is the next stage of the data analysis for kind of this data set. But interestingly, you still see this net contraction. So there is a net effect on trunk dynamics as the season progressed and as we added more lanternflies. So then when we pivot and we think about metabolism and primary metabolites in terms of nitrogen and carbon, when I say carbohydrates, I mean soluble sugars and starch. For the sake of this presentation, I only have starch represented, but again, the lanternflies are very intensive, heavy feeders. So I also thought that we'd see this density dependent response such that, the higher lanternfly infested vines would have a stronger reduction in both carbohydrates and nitrogen. And then when we looked at that and looked at late season starch and nitrogen concentrations, we saw mixed results. So this is the starch for late season tissue concentrations. On the left is the stem grouped together, and these are root starch concentrations. And you see no difference really in the stem, but then we did see a significant reduction with the medium vines for roots, and then a non-significant reduction for the high vines. So I thought this was really interesting because this implies then that the vines are allocating less starch, or less starch is being stored in the roots in response to lanternfly feeding, which is an interesting thing to know, especially because as we think about the next couple of slides, which I'll transition to now, are nitrogen data. And when we look at the nitrogen concentrations, again, this is percent nitrogen. These are the leaves clustered together, stems, and then roots. And again, it's important to note that even though this is late season, these leaves were sampled pre onset of senescence. And we see a reduction in nitrogen in the medium and the high treatments. We don't see any reductions in the stems. And then again, root specific reduction, but only in the high vines this time. So we're seeing these reductions in starch and nitrogen in the roots, and which is interesting and has potential implications because that's where a lot of these compounds are stored for use for next season's growth. So that really started getting us thinking and wondering, well, the lanternflies being phloem feeding, are they doing something that's blocking translocation from occurring? So these compounds actually aren't even being allocated or able to be stored in the roots because of some kind of blockage. And interestingly, we observed some things during the season that maybe kind of facilitated our thought towards this. The first being that when we took samples of stems that lanternflies had fed on and removed the bark, we tended to see these striations on the phloem itself. And we didn't know if this was just simply cell damage due to lanternfly probing or feeding, or if they were callose depositions in response to phloem damage from feeding, or what exactly. That's something we'd have to look at further, but we only saw these in vines that were infested, which was interesting. And then in some growers in Southeast Pennsylvania, from some red varieties, some areas that were highly infested started expressing this kind of red-leafed symptom. And we do see this in grapevines in other scenarios, namely in heavy and vines that have heavy loads of grapevine leaf roll associated virus, which resides in the phloem and blocks the phloem. And when that happens, vines that are heavily infected tend to accumulate more sugars in the leaves that then lead to this anthocyanin production and this reddening of the leaves. And it's essentially a photo inhibition mechanism. So we were wondering, seeing this, well, maybe lanternflies potentially are blocking, whether either physically blocking the phloem or doing something else that's preventing translocation and leading to a photo inhibition effect itself. And this would be really nice if we could parse this further, because this would give us a neat mechanism for how potentially the lanternflies are even reducing the allocation of nutrients into the roots if translocation is being blocked, as well as a nice corollary to these reductions in photosynthesis and gas exchange parameters that we saw. And to measure this, we took samples one of my colleagues, Lauren, took samples pre-dawn of all the leaves, and we extracted soluble sugars from those. These are from the red leaves, as you can see, and these are from the green. And I'll be analyzing those statistically soon. That was something I started way in the beginning of this year, and then the pandemic happened, and it's kind of been hard to get back to it. But we're hoping to do that soon, because that would really give us some insights into a potential mechanism of what the lanternflies are doing. So then, we have these results and these other questions, but what about the carryover effects that I mentioned? And we mainly focused on three aspects of this: vine nutrition, shoot growth, and leaf gas exchange. And again, I thought we would see a density-dependent response in the nutrients, as well as the shoot growth and the leaf gas exchange. And interestingly, that wasn't the case. So this is shoot growth data taken by Lauren again, who did most of the measurements for this question. And what you see is this is the shoot growth rate across June into early July, which again is the most intensive period of shoot growth for grapevines. I mean, like 2.5 centimeters up to 5 centimeters a day is how quickly they're growing. And no significant differences seen here, no treatment effect. But it is important to note we had a very heavy frost, late spring frost, that damaged a lot of the shoots and the vines. So the shoots we selected for this measurements, we selected after the frost. And we tried to standardize them as best as we could and taking into account the damage that we saw on choosing healthy shoots. But it's still important to note that the frost may have potentially obscured effects, which is why it's important to take multiple years of data, and which is why we're going to be repeating these measurements next year. And it's also important to note too, when I say 2020 shoot growth, this is not a different experiment. These are just data taken from the same vines that we infested last fall. So follow-up data points. So no effects on shoot growth. And also I didn't see any effects on gas exchange as well for any of the parameters. So they seem to be equal across the board. But then when we looked at the nutrients, we specifically focused on macronutrients, and we didn't see any effects there with the exception of phosphorus. And we took these measurements at bloom and took petiole samples, which is typically what's done for grapes. And we were kind of puzzled by this. We didn't expect to see this. And we also didn't see a treatment density dependent response. We just saw a flat treatment response. So I don't know if this is potentially something related to lanternfly specific nutritional need or something else, but it's exciting because it opens up another avenue of research in terms of looking at, more nutritional responses to lanternfly feeding. So this was really nutritionally the main difference that we saw. Everything else, nitrogen, potassium, was the same. So to kind of overview the results of what we saw in our hypotheses, again, I thought we would see this density dependent response for the physiological parameters that we looked at. And we did see that for the leaf gas exchange for carbon assimilation, leaf transpiration, and stomatal conductance. But again, we didn't see that for the sap flow or the dendrometer data. Instead, we saw no difference between the low and the control vines, and instead a grouping of the medium and high vines in terms of reduced sap flow as well as greater trunk contraction. And then in terms of our primary metabolites, again, density dependent response with increasing responses with increasing lanternflies. And we didn't see that again. Any starch reductions we saw were a little inconsistent, but more tissue based, namely with roots, with again, significant reduction in the medium. And then in terms of nitrogen, we saw foliar responses only in the higher lanternfly treatments, as well as with the roots. And then as far as carryover effects go, we saw, again, that decrease in phosphorus, but no other effect there. And then in terms of shoot growth and leaf gas exchange, no effect as well. But it's important to say that this is only 1 year of data. As you know, we're growing plants in very variable weather conditions. We tend to get really severe winters some years, not so much other years. And this past winter was extremely mild. We did have the frost event, but it's likely that these results, or possible that these results, could be different if we had a different type of winter. So on that note, we did repeat this project in 2020. Here's a photo of our experimental design that you can really see all the cages up and down the vineyard rows. So we repeated the experiment in 2020 using 16 different Riesling vines, same vineyard, but different Riesling vines, because while we're interested in cumulative repeated infestation effects, we're trying to validate the data from 2019. So we needed a new kind of crop of vines. But then we shifted our treatments a little bit to do more spotted lanternfly densities and to try to regress the data set and see, if the relationships differed over more density. So we did 16 different treatment densities ranging from 0 lanternflies to 16 lanternflies per shoot. And then, as I mentioned, these measurements will be used to validate our data from last year. And then we also took some measurements on more plant defense related responses to try to begin probing that pathway. So that's kind of managing and looking at this data in addition to the other dendrometer data is really what's the next steps for this project. So then just to I give a shout out to everyone who's helped on this project. Again, like Michaela was saying, this has been intensely collaborative, and there's no way this project would have happened if it was just, one of us. And every single person has had such a integral and important role. And big shout out to Don Smith, who actually is the architect behind all of our cages and really helped facilitate the whole experiment, as well as our collaborator grower, who has been just incredible, and all of our funding resources. Yes, that completes my presentation, and I'm love to take any questions.
Thank you, Drew. Does anyone have any question for Drew? You can unmute yourself or I guess use the chat. We have a few minutes.
Yeah, well, Drew, Armin here. So, yeah, why did it seems that photosynthesis went down? Why?
That's, yeah, I think, I think that's one of the big questions, especially because with the high intensity, there was a really strong reduction right away. And they're not damaging, the physical leaves themselves like a chewing insect would. So what I think may be happening could be either some kind of, whether it's some kind of other like stress pathway related response that the lanternfly is triggering, or maybe potentially through the nitrogen it's removing, it's having some indirect effect on leaf chlorophyll content or photosystem functioning. And that's something I would like to pursue further and is a little bit more down a different road. But yeah, that's a great question, and I think is something we'd like to look at further. And it's been a topic of much discussion.
So, Drew, in the field, are growers seeing a really big significant difference in their yields with the lanternflies around or not?
That's a good question. I wouldn't know as well as maybe our extension agent who works mostly on this, but I think there are visible reductions being seen, and in some cases vine death. I mean, we do know growers that have lost vines following intensive feeding seasons, but it's also really hard though too, because again, vines respond so much to there's so many different weather conditions and other aspects that could be contributing to things, that it's hard to, just to like pin it directly on the lanternflies themselves. But it's something that we're trying to parse out. But I do think there are, yield reductions being seen, but that's something too that we would have to further investigate ourselves and actually set up a trial, looking at, actually incorporating that further into an experiment.
Another question I had was, so you had, 6 different cycles. Are growers actually spraying 6 or 8 times a year for these things right now?
Yes, my understanding is that, growers are spraying a lot, which, is economically costly. And it's also led to some other issues and problems too with secondary outbreaks of other pests, as well as, some more other ecological effects on like local insect populations. But yeah, growers are spraying, and this is in the vineyards that are having really heavy infestations, it's pretty constant spraying. So there's a lot of interest in, trying to reduce that, because also you reach a point too where you can't spray anymore with the pre-harvest interval, and you're kind of in a tough position.
And there is a question in the chat. I don't know, do you have access to the chat?
Yeah, I can see it. Thanks, Imperna, for that question. Yes, that's something. So Simperna is asking if there was honeydew-induced sooty mold growing on the leaf tissues that could have interfered with the photosynthesis. And that is something that other people have seen, like really intensive sooty mold as a response to the honeydew from the lanternflies that they produce. Because I mean, when they're really feeding and when there's a lot of lanternflies on a given plant, there's a lot of honeydew and potentially a lot of sooty mold. But we didn't see we didn't see a lot of sooty mold. We saw some here and there. And I actually went and measured gas exchange on sooty mold leaves as well. And that doesn't seem to be, we don't think that's the mechanism for reducing photosynthesis, at least for our experiment, because there just wasn't enough sooty mold to really cover the leaf that much. Compared to other, instances where like the leaves are just like black. We didn't see anything like that.
So, and did you, Armin, again here, another question. I can get the, what happened with the, let's say the measure of transpiration, or if you try it, with the contraction and expansion, the diurnal cycle of the diameter of the stem.
Mhm.
Did you see a reduction in transpiration?
Oh, in the diurnal. Yeah, see, that's something I need to look at further. It's been a while since I've looked at that individually calculated, like the peak to like the highest to the lowest point. I didn't think there was a noticeable reduction that way. They still seemed, even the really infested vines still seemed to respond pretty closely to like environmental cues and what we would expect with like increasing sunlight and increasing temperature. And so they were still like tracking pretty well to diurnal weather conditions. But that's something I'd have to investigate further and really like kind of test out in R. Yeah. That's kind of one of the next steps.
So, but the, photosynthesis went down, but stomatal conductance went down apparently, right?
Right.
So transpiration must have gone down.
Yes, yeah, it did. And with the high vines, and that's some of the data we see this year. So our data from the sap flow kind of validates some of what we saw last year. And we do see, even in the middle of the day, a reduction in transpiration. Transpiration's still occurring, but just to a much lower level. But so you would expect to see that concurrent diurnal change in the dendrometer too, but I would have to look at that more because I think there's also an interplay between the phloem and the xylem and water movement back and forth there. That kind of complicated the data when I was looking at the 2019 data. That didn't give me a really clear idea of what was happening.
All right.
Okay, I think it's 1:12. And if there are no more questions, let me see, there is one. Okay, it was Sampurna saying thank you. Oh, no, it was, are you looking at ABA accumulation?
Oh, yeah, Sampurna, we are not. We thought about doing more hormonal kind of sampling, but it just was too difficult. But that is something that we could potentially incorporate into future studies. Because we're looking to do some potted vine experiments looking at things. Just in the field, it just would be too difficult to sample, I think.
Okay. I think thank you, Drew, for the update and presentation. We can virtually clap, yeah. And I think, and so Rob left, so I think I can just stop the recording, I guess.
Yeah, I think so.
Okay, and hopefully, and it will be fine, right? I've never done this. Okay, it will be fine. Okay, thanks everyone.
Yep. Thanks, everybody.